So first phase of mitosis is prophase. The DNA is going to go from all unwound and loose to tightened up. Let's say that for a lab, what I wanted to do was I gave you all 92 strings that were a meter long each. And there were 23 different colors that you had to deal with, but I took all those strings and I mixed them all up and I put them in a pile and I gave them to you. And you all, I took those strings and I wound them around something, and you had 92, basically like your strings all organized into nice little sticks. Who's going to separate all the colors into groups of 4 first? The unwound strings or the wound up strings? The what? Unwound, they're all mixed and they're like, oh, yes. So the wound strings, right, all you have to do is like grab the 4 yellows and grab the 4 reds and grab the 4. And that's essentially what's happening here, is that DNA is all like unwound and it's all like mixed together in the nucleus. And in the process of prophase, all the DNA is starting to wind up, condense, and become easier to move and organize around the cell. So that, remember, at this point, the DNA is replicated. So if we're talking about a human cell, that we have our original 46 chromosomes and they each have a sister chromatid. So you've got 92 pieces of DNA that you have to organize in the cell. Now, remember also that you can't just choose any 46 and move them over here and any 46 and move them over there. You've got to choose a pair of sister chromatids of number 1 and the other pair of sister chromatids of number 1. And you've got to put them together, and the number 2s, and one of each number 2, and one of each number 3, and one of each number 4. So you have to have some very strict organization here so that each of your 2 daughter cells gets a pair of number 1s, a pair of number 2s, a pair of number 3s, all the way down to your sex chromosome pairs. Okay, so it's got to be very, very, very organized and well orchestrated to do this. And that's what prophase is setting up. We're setting that up. Also, if you've got this amount of space in your cell, but your nucleus is this big, wouldn't it be better if you kind of like dissolved your nucleus envelope and then you could use the whole of the cell to move all of your chromosomes around? And also you're going to, put a set of organelles here and a set of organelles there. So you're going to dissolve the nuclear envelope also to give you more space in the cell, to make sure that each cell is going to get the right set of 2 number 1s, 2 number 1s, 2 number 2s, 2 number 3s. We have these microtubules that connect to each chromosome, each sister chromatids pair at their centromere where they're connected. And that, like those strands, it's called spindle. It's going to push them and pull them so that we get a whole set, a full genome of the right kind and a full genome of the right kind. The right 46 here and the right 46 here to divide into 2 cells. So our nuclear envelope is disintegrating temporarily. Our DNA is condensing from chromatin into chromosomes and really specifically into sister chromatids. Our spindle is going to form. We have these 2, what they're called centrioles. They're a pair of organelles that go to the top and the bottom of the cell. What we call that is the poles of the cell. Just like you have a north pole of the earth and a south pole. Poles mean each side. So that those centrioles are spitting out, or they're making that spindle. That's going to push all the chromosomes to the middle and separate the sister chromatids to get a full set of the right kind on one side of the cell and a full set of chromosomes of the right kind on the other side of the cell. What we see, yeah. They're already in the cell. Yes, they're already there. They don't really, this is like when they shine, when they do their big job. Yeah. So I went over all of this. What the cell is going to look like under the microscope, and this is a little dark, but prophase so we'll kind of keep a running tally. That interface is going to look like this. It looks like a somewhat solid nucleus. And prophase is going to look like this. You're going to start to see chromosomes changing from chromatin to chromosome. The next phase is prometaphase. Prometaphase, the spindle fibers are fully formed. It's going to look somewhat like this, where you can see the actual chromosomes themselves. Each of these is really indicating a sister chromatid pair. They used to call this early prophase and late prophase, but now they say prophase and prometaphase. In the details of the cell, if we had like a really strong electron microscope, we would see more details like this, where you would see that each of your chromosomes has their genetically identical sister chromatid in that centromere region. They are connected. They all start lining up in the middle. You have the spindle fibers fully formed. Sometimes you can see kind of like, it might look like this, or it might look like you see some strands coming in. Depends on the type of cell we're looking at. But you do start to see individual chromosomes. Okay, metaphase. Think metaphase, middle. That spindle's pushing all the chromosomes, the sister chromatid pairs, to the middle of the cell. What you're going to see is a dashed line in the middle of the cell. It's showing you all of those chromosomes, what their sister chromatids are in the middle. If you've got a good microscope, you'll see the spindle is fully formed. So your metaphase looks something like that. Metaphase, middle, your chromosomes are in the middle of the cell. You will see the orientation, which is shown well in the illustration, that the pairs, each of the sister chromatids, are lined up horizontally, because what is going to happen in the next phase is that they are going to divide at their centromere, or their nucleosomes are going to divide. And so that one chromosome of each kind will go this way, and its genetically identical sister chromatid will go the other way. And so they're going to go split, 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 split. And so that you get a complete set of each kind on one side of the cell and the other side of the cell. So the orientation is really important here. So anaphase, sister chromatids separate. They go to opposite poles or sides of the cell. Polar microtubules are orchestrating this by making the cell longer, stretching it out, so that we can make two not only equal amounts of DNA on each side, but a set of organelles, and then also enough cytoplasm for the new daughter cells to function. So these polar microtubules are stretching that cell out. So our anaphase, what you see is you're going to see a line of DNA on one side and a line of DNA on the other side. Here, you can see it's darker where the chromosomes, those sister chromatids, have been pulled apart to become individual chromosomes, one set here, one set there. You can see here also the spindle pulling one set this way and the other spindle pulling the other way. You also will see an anaphase that the middle, what we call it, this imaginary metaphase plate. Metaphase plate means like all the chromosomes with their sister chromatids are lined up, and you see that line. It's not real, it's just kind of gives you an idea of the middle, the metaphase plate. It's an imaginary thing. There's nothing in the metaphase plate, an anaphase. And you see a good spindle pulling. You can see the cells getting elongated, getting pulled longer. In telophase, we kind of get rid of everything. We get rid of the spindle. We get rid of, so we get rid of the spindle. The DNA is going backward. It's going from chromosome to chromatin form. Nuclear envelope is going to encapsulate our DNA on each side. You're going to get a continuous pulling of those polar microtubules. Our microtubules in general, our spindles starting to go away. Our cell is nice and long so that it can divide into two. So this will give you an idea of what you're looking for under the microscope. In our cells, because we don't have a cell wall, we'll start to see a pinching in telophase. So your telophase is going to look kind of like that or that. Both of those are telophase at the beginning or end. You can see here it's shaped like an 8. Here you can see still some of the spindles around, but it's starting to divide into 2. It's fully pinched in here. And then our last part of the mitotic phase is cytokinesis, cyto for cytoplasm, totally dividing up the cytoplasm to make our 2 new cells. And it's just a progression from our telophase pinching in to completely separating. And what it's going to look like is that you have two cells in interphase right kind of smashed next to each other. So this phase and this phase are very similar. Right, what we started out with originally, our cell there. See our nucleoluses. And that's it, cells divided. So let me talk a little bit about the difference between, oh no, before we get there, let's have a question. So which of the following statements about the chromosome of prokaryotic and eukaryotic cells is true? Let's go through these each. So both prokaryotic and eukaryotic cells have multiple chromosomes. Do prokaryotic cells have multiple chromosomes, not including plasmids? No, they have one chromosome. So this is not true. Prokaryotic cells have one chromosome. They have other pieces of DNA, those plasmids. They do not count as chromosomes. Do they, say it again, so prokaryotic cells, do they what? Oh, B. Chromosomes of prokaryotic cells contain a circular DNA double helix, but the chromosomes of eukaryotic cells contain linear DNA. That sounds pretty good to me. But let's read on just in case. The chromosome of prokaryotic cells is present in the nuclei. Do prokaryotic cells have a nucleus? No. So, nah. Chromosomes of eukaryotic cells are attached to the plasma membrane? No. But the chromosomes of prokaryotic cells float free in the cytoplasm? No, neither of these is true. So, yes, B is correct. Okay, let's look at the difference between a cell that does not have a cell wall and a cell that does have a cell wall. And what happens when we get into our telophase or our late anaphase area? How do the cells separate from one another, the daughter cells? The stages, all this is exactly the same. We just have a different barrier when we have a cell wall, kind of like another step, because cell walls are very rigid. So we're going to see the process of the separation of the two daughter cells a little bit differently. All right, animal cells. It's called the cleavage furrow. The two cells start to pinch in. In our societal terms, think about what the term cleavage means, right? Cleavage looks something like a pinch in. We see it. Maybe you'll start seeing it in anaphase, but definitely in telophase. Turn this on its side and think about that as cleavage. It'll give you an idea of where the term cleavage and for women comes from. Yeah. Nope. Cell pinches in, you get two genetically identical daughter cells. This is what our cells really look like, with all of the little attachments that come off of the cell. Our cells actually look more like a tennis ball. They look fuzzy. If we had a really, really good electron microscope, we would see the details of all the little markers that come off of our cells that communicate with other cells. They're not flat surfaced like this. But you can see the pinching in of the two cells together. Okay, plant cells, fungus cells, any protocells that have a cell wall, they have to not only have a plasma membrane separation, but also a cell wall separation. We have what's called a cell plate, forms between them. In our animal cell, which is more pliable and only has a plasma membrane, this can easily pinch in. But what we have happening here is that you've got to have the plasma membrane starts to pinch in, but also you have to make a new cell wall called a cell plate. What we usually see under the microscope is you will see a dash during telophase in any cells that have a cell wall. So this here is the cell plate. What you're looking at, the difference between an anaphase and a telophase is this. That cell plate will tell you that is telophase in a cell with a cell wall. So here you can kind of see that through illustration, but it really looks like not a complete dash across. Telophase, you're going to see a little dash in the middle. Okay, so you sprain your ankle. Platelets are attracted to the sprained area, something that's going to clean it up. Why do they release platelet-derived growth factor and epidermal growth factor, which activate the cell cycle? So let's take a look. The skin must grow to compensate for the swelling. The skin going to grow. Maybe. I mean, let's put that on hold. The growth factors must destroy the damaged tissue. Maybe. The damaged cell must be replaced through mitosis. Does that sound like something we've been talking about? Specific terms like mitosis, right? So this should give you a clue. Even though these sound like, they kind of sound like they could be right, this one clearly has a term in it that we've been talking about. And we did talk about that the function of mitosis is growth, repair, development, and maintenance. And so what we know is that if something, it's got all these fancy terms in it, right, this question, but we know that something is destroyed, so we have to repair it, and we do that through mitosis, that this, even though these two are maybe precarious, you're like, they sound really smart. Don't choose them. Choose the one that makes more sense in terms of the content that we're looking at. The damaged cells must be replaced through meiosis. If you're like, shoot, I can't remember the difference between mitosis and meiosis, remember, meiosis is only for making sperm and egg. Are sperm and egg going to come and fix our sprained ankle? No, right? Okay, so C is the best choice. Yeah. Your body cells are going to use mitosis, like your toes, to get fixed. So these are just like, I like to go through questions like these just to give you ideas. I know all of you are headed down paths where you're going to have to take these higher level multiple choice exams. And so I like to kind of like go through just kind of like test taking strategies to look for. Okay, there's checkpoints. Between many parts of the cell cycle, we have things that happen, we call them checkpoints, that make sure, did what we just did go perfectly or near perfect? Let's check that out. That's a checkpoint. Kind of like going through, like if you travel somewhere, customs. You got a checkpoint. They got to make sure you are who you say you are, and then they stop you and they look over your information and say, yep, everything looks good, move on. That's like what the cell does. Imagine that your textbook, well, it's digital, right? So your digital textbook is, for this class, is probably about 100 and, let's just say 150 pages for this class. To go through the process of mitosis to make 2 new daughter cells, what our body has to do, our 46 chromosomes, let's say that they contain an equivalent amount to 10 copies of our textbook. That's probably close to the truth of the amount of information that is in our DNA that has to be replicated. Do you think that a cell, let's say it takes 1 day, 24 hours for a cell to go through the process of mitosis, and it has to copy 150 pages 10 times, so 1,500 pages of very high level information in 24 hours. Just like I said before with our packet with the prokaryotic cell, we said the prokaryotic cell in 20 minutes is probably going to make mistakes on copying this, right? Do you think our eukaryotic cells are going to make mistakes in copying 1,500 pages of information in 24 hours? For sure. Yeah. So we have these checkpoints to kind of check things over. There's 3 different checkpoints. Between G1 and S, we want to make sure, did replication go well? So that's like your spell checker, your grammar checker. They're going to make sure it goes pretty good. Were all the chromosomes copied the right way? Did any of them stick together? Did number 2 get ripped in half and is attached to number 1? G2 to meiosis checks that out. Metaphase to anaphase, did they separate appropriately? Did the sister chromatids come apart, or did some of them stick together and 2 sister chromatids move to one side of the cell when we should have separated those sister chromatids? That happens between metaphase and anaphase. Is it likely that some mistakes are going to get through these checkpoints? What do you think? I think sometimes, especially this one, do you think a spell checker, grammar checker is going to work perfectly on 1,500 pages of information all the time? No, because there's things like, just in our regular in English language, right? We have there, and there, that you have a lot of different ways, or to, and to, and too. There's a lot of different ways to communicate a similar sounding word in different ways. So those mistakes we know get by, right? If you always just rely on spell check and grammar check for your paper without reading it, sometimes mistakes still get through because of little like duplications of similar terms. So yeah, we're going to get some mistakes that go through. The thing is how bad are the mistakes that get through will determine the future of the cell. Will the mistakes that get through, will the cell be able to function with those mistakes or not? So our checkpoints are here, here, and here. That we do have them, but sometimes mistakes, mutations get through. Okay, so G0 phase. Here's the thing about that G0, sorry, we didn't talk about G0 phase. G0, we talked about G1, S, G2, but we didn't talk about G0. There's what is called a G0 phase. This is a quiescent phase. The term quiescent means that the cell is not active at that time. Depends on the kind of cell, how long they stay in G0. Some of them are doing nothing until they have to do something. Like, for example, your pancreas cells, I've been talking about islet cells that make insulin. In your pancreas, you have specialized kinds of cells called islet cells, ISLET. They make insulin. Do we want to be making insulin? Do those cells want to be making insulin 24 hours a day? Would it be economical for that cell to just pump out insulin constantly if we don't need it constantly? When do we need insulin? Does anybody know when we need insulin? Yeah, when we eat. We need insulin when we eat, because what insulin does is like a facilitator who takes sugars and other nutrients, other energy sources in our blood, and it helps them to get into our cells. But we don't do that 24 hours a day, so we don't need insulin 24 hours a day. It would cost us a lot of energy to constantly be making insulin. So our islet cells go through this like G0 phase where they're not producing insulin, and then they are. And then they're not. They go back to G0, and then they are. And so our pancreas cells, our islet cells, are back and forth throughout the day. There are some cells who may only need to be active, like at puberty, for example, that they're in G0 for the majority of your life, And then puberty hits and they're on. And they're on for maybe like 5 or 6 years, and then they go back to G0. So we have different cells when they're active and not. They stay in this G0 at different times. Some are all day, some are different times of the day, some are with the menstrual cycle once a month over the course of a week, some are only doing during puberty. So there's different ways that our cells spend in G0. Did you want to say something? Wait, say it.
So access a facilitator to glucose slave transport. So the vesicles.
Okay. Okay. Let's talk a little bit about positive, negative cell cycle regulators. We have these different cell regulators. They help the cell through the process of division. Our positive regulators, we have two that are called cyclins and cyclin-dependent kinases, CDKs. They fluctuate in their activity during the cell cycle. What they do is they regulate the checkpoints. They kind of like say, hey, checkpoint 1, you're on. Hey, checkpoint 2, you're on. Checkpoint 3, you're on. So they're making something happen. That's why it's called positive. They're helping out the checkpoints to check out. Did everything go okay at these 3 different checkpoints? So, for example, our cyclin and CDK, when they are not working, they're not connected. You can see they're separated. When they are about to work and help out the checkpoints, they bind to kinase, they bind to each other, and they make the proteins that are the like checkers for the checkpoints, they make them function. When they're not working, they go back to being separated. Negative regulators. All right, let's say that the last step did not go well. That the checkpoint, one of those 3 checkpoints says, oh, things went really, really badly. And the checkpoint that I had to check out all the information, these kick into play. And essentially what we're talking about is negative regulators. When a checkpoint says, this is bad, we cannot let this cell move forward and do its job because it's a really bad situation. So what they do is they go through a series of steps to help the cell kill itself. What that is called when a cell needs to kill itself because things have gone disastrously wrong, it's called apoptosis. Programmed cell death. It says this cell's in bad shape. Don't let it go forward. No, just this basic stuff. P53 works with the first checkpoint. It's making sure that the DNA, well, if the DNA is really, really damaged in the copying, it's like, let's induce apoptosis. P21, it's going to check over the CDK cyclin complexes. It also works with P53. The 3 of these, the last 3, P53, P21, and the RB, they all work back to, I should say P21 and RB all work back to P53. If their job says this is bad, they tell P53, kill off the cell. So it's just a series of different communicators and what has gone wrong with which checkpoint. And then RB, retinoblastoma. Again, what it's going to do is it's going to check and see that everything has gone correctly, and it's going to go back and tell P53, kill it off. And this is, again, back to the G1S transition. It blocks that. For these, I just really want you to know what the positive ones do. They help the checkpoints to act. And these 3 negative ones, if something goes wrong at a checkpoint, they say kill off the cell. That's it. So just to simplify the objectives here, just knowing the positive ones help the checkpoints. The negative ones say it's bad, kill the cell. All right, so very general. This is just an introductory class. So again, we're just generally speaking about different things that happen in our body. Cancers. There's thousands of different kinds of cancer. If somebody's going to say, I cured cancer, you'd be like, which kind, right? Because there's so many different kinds. And all the different kinds of cancers work in many different ways. In general, cancers, what it means is that the checkpoints, they just keep saying, like, go through, cell. Okay, let's go again. Let's go again. Let's go again. There's no G0 phase where the cell is like off and it's not doing anything and it's waiting to do its job. It's just saying, oh, mitosis happens. Let's go again. Let's go again. Let's go again. And so the whole cell cycle in cancers and generally goes around and around and around. Different cancers go at different rates. Different cancers are activated on different genes, different external factors. There's a million different reasons why even a single kind of cancer might work in a certain way with different, again, genetics, that most of the time when we're talking about a kind of cancer that someone is susceptible to, you have a genetic predisposition. What that means is that there's something in your genes that say you might, given these circumstances, get this kind of cancer. So, for example, biological females who have XX, because we don't have a Y chromosome, and we're talking about biological females who have 2 X's and 2 X's only, we don't have a Y chromosome. So we are not susceptible. We do not have the genetics to get testicular cancer. We do not have the genetics to make testes, right? So we can't get that kind of cancer. We're not susceptible to that. Men have those genes, and given environmental circumstances, they have the possibility of getting that kind of cancer. So this is all just very complicated. In general, we have what are called oncogenes. They're mutations to genes that are part of checkpoints or different parts of the cell cycle that allow the cell to just go, 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 replicate, 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 replicate. The cells are programmed to ignore the checkpoints and go through them, and go through them. The checkpoints, something malfunctions there where the cells are like, wait, didn't we just replicate? Oh, well, let's go again. Even though they don't need to. Something malfunctions in the checkpoints that allows them to go through. Again, thousands and different kinds and reasons and genetic factors and environmental factors that cause cells of different kinds of cancers to push past. Some cancers are due to mutations to RB, P53, and P21. Remember, these are negative regulators that notice something's bad. The cell's bad. Kill it. They're mutated so that they can't recognize when something is bad. And so it makes the cell just go, 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 go. Again, simplistic terms. Cancers are very complicated, just to give you some ideas of what we've been talking about. And what we see for many of them, the checkpoint enforcer is mutated. It doesn't work. And it could be checkpoint 1, checkpoint 2, checkpoint 3. It could be our tumor suppressor proteins. It could be our positive and negative regulators. It could be a million other things. But in general, for example, the checkpoint enforcers don't exist in cancers like, and again, some kinds of breast cancer, lung cancer, brain cancer, pancreas, bladder, stomach, colon. But there's a lot of different kinds of each of these too. Lung cancer, there are certain kinds of lung cancer that are due to this, and you could be a person who's never vaped or smoked in your life, and you get lung cancer because it's a certain kind of lung cancer related to your checkpoint enforcers don't exist. They're mutated. There's other kinds of lung cancers that are more environmentally caused, somebody who smokes or vapes their whole life, and they get a certain kind of lung cancer. So again, just a lot of variation. Yes? Mm-hmm. Yeah. Wild. Yeah. So here's how it kind of like normally goes, that your checkpoint regulators will notice that the cell is damaged and you go into apoptosis. That's what should happen to a damaged cell. When we're talking about cancerous cells, this damaged cell, there's a mutation here that doesn't cause apoptosis, and that damaged cell, it replicates. Cancer cells have different kind of effectiveness levels. This is very generic again, that let's say you've got the first cancer mutation of a certain kind, these replicate, cause a second mutation of the kind for the cell. It just regulates, you don't get the full-on cancer yet. A third mutation occurs, you don't get the full-on cancer yet, but it's a fourth mutation of that original damaged cell that causes the cancer to accelerate. All right, let's talk about treatments for cancers. There are so many different kinds of cancer treatments. Even within a certain kind of cancer, there are different treatment regulations. A lot of this just depends on what genes are mutated and cause that cancer. So many great advancements now. If you are somebody who's really into solving puzzles, research in different kinds of cancer are starting to customize that if you have, like for example, my best friend, she had CHEK2 positive breast cancer, and it's related to 3 different genes. And for those 3 different genes, she was negative, negative, negative for all 3. 10 years ago, the research that was done said somebody with the 3 negative genes, it's a death march, that you will die of this cancer. We do not have a treatment, but we can do things to sustain your life as long as we can. But she had it 8 years ago, so let's say 18 years ago, when she had it, there was advancements in the research on this kind of cancer that somebody with these 3 negative genes, they had a great protocol, and now she has been cancer-free for 8 years. Cancer-free, not maintaining a life of having chemo constantly, which is what her mom had to go through. Her mom was on chemo for 20 years, but now she is cured. So there's a lot of, in terms of all of these like inhibitors and working with genes and things like that, there's so much you all can do to solve puzzles that can elongate and save people's lives. So if you're somebody who was like, I want to be a pharmacist, and you think about like what that means is you're going to go work in CVS and you're going to take orders from doctors and you're going to fulfill those orders for your patients. You can do so much more with a pharmacy degree now than ever. It's really exciting times. Many kinds of cancers, you take anti-cancer medications that kill off fast-growing cells that go through the checkpoints, go through the checkpoints. These drugs, these anti-cancer drugs, target cells that are going fast. Chemotherapy, we call it. So when someone's on chemotherapy, side effects occur because we have not only your cancer cells that are growing quickly, pushing past the checkpoints, but we have a lot of other cells in our body that go through checkpoints constantly because they replicate constantly. Like our hair, our follicle cells grow. So someone who's on chemo often will lose hair on their body. They lose their eyebrows, their eyelashes, their head hair on their body. Because the chemotherapy drugs are targeting quick growing cells, cells that go through the checkpoints constantly. So hair loss, our digestive system cells are constantly being replaced because they are highly taxed. They are worked hard. Our stomach cells, they are exposed to food, which can be jagged. They do a lot of churning. The layers of our stomach get sloughed off or pulled off with digestion. And so we have to constantly be replacing our digestive system cells. Oftentimes people with chemotherapy, they will lose their hair on their body and they will have very bad digestive system issues. But not always, and it depends on the kind of cancer and again, different checkpoints that are focused on. I'm sorry, say it again. Probably, yeah. Let's answer this question. Which of the following is not a function of mitosis in a multicellular organism? It's not. We're looking for the one that is not for mitosis. Okay, which one do you think? So it's okay. So growth from a fertilized egg. So what we mean is that a fertilized, remember, fertilized egg is a zygote, right? So that one is like sets you up to go like, you got to really know your terms. So fertilized egg, that's the toughest one. Fertilized egg, it is the fertilized, meaning the sperm has come in. You've got the zygote, and that's ready to go through mitosis. The job of mitosis is growth, repair, development, and maintenance. So those are true. Production of gametes is for what? Meiosis, yes. Okay, so on to meiosis. Meiosis is different than mitosis. Mitosis, we had one set of phases, right? We said we had interphase, prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. We do that once, we're all done. We have two sets of 4 phases for meiosis. So we're going to go around one set and then set number 2. A lot of very unique things happen in meiosis 1 and here, especially at prophase 1 and anaphase 1. Okay, so meiosis 1, let's talk about prophase 1. Similar things happen as what has happened in mitosis. Nucleolus disappears, nuclear envelope fragments, the DNA goes from chromatin to chromosomes, centrioles migrate from the poles, they go to 2 different poles, spindle starts to form. Yeah, that all happened. All of this happens in mitosis, but our chromosomes do freaky, weird, cool things. We get our sister chromatids. Remember that in mitosis, that our number 1 sister chromatids lined up, and our other number 1 pair of sister chromatids lined up. They did not interact with one another. The number 1s lined up, the number 2s lined up, but they didn't interact. Here, what's happening is the 1 sister chromatids and the other 1 sister chromatid pair, they come together as a group. So we've got 1, 2, 3, 4. Tetrad means 4, because you've got a pair of number 1s replicated sister chromatids and the other pair of number 1 sister chromatids. So 1, 2, 3, 4 come together, and they undergo what's called synapsis. Yes, correct. What they're going to do in this synapse is that they are going to exchange parts between chromosomes. This is weird. In synapse, when they all come together, 1, 2, 3, 4, they will cross over pieces of chromosomes, chop, chop, and exchange. So it's like if I went up to my friend here and I was like, I'm going to take your arm, I'm going to chop this one off, I'm going to chop mine off, I'm going to put my arm on you and your arm on me. That's weird. It's going to make new genetic combinations, right? He's going to have the genes for my arm on him, I'm going to have his genes on me. That's weird. Weird. Cool. It makes new combinations of genes that will occur in the sperm and egg. This is one of the reasons why, if you are a person and you have a sibling, why you don't, and let's say that you are a female and you have a female sibling, this is why the two of you are not exactly alike. It is why you have some similarities and differences. This is one of the reasons why. Because the chromosomes from your biological mom and your biological dad chop, 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 chop each other up and exchange. That's weird and cool. So this is the start of synapse, is that the number 1 chromosome with its genetically identical sister chromatid and the other number 1 with its genetically identical sister chromatid, the 2 original number 1s with their genetically identical sister chromatids, they come together 1, 2, 3, 4 as a tetrad. In mitosis, they did this. They did not interact. They lined up in the middle. They didn't care what this pair of number 1s chromatids did not care what this pair of number 1 chromatids did. They minded their own business. Here, they get together. So first step, they get together. Second step, they use proteins and they zip all their parts together. Zip, zip, zip. Chop, chop, exchange. They exchange equal parts so that the gene batting pattern stays the same for each. One doesn't get like a little bit and the other one gets a lot. It's equal amounts exchanged. So that on the different genes at different places, the different loci of the chromosomes stay the same. Equal parts are exchanged. Just like my arm, we cut at the same place and we exchange. So here, what you see in prophase 1 is you see that these kind of chromosomes line up in a tetrad. This is a different kind. Let's say these are number 1s. These are number 2s. The number 1s get together in a tetrad and undergo synapse. Number 2s get together in a tetrad and undergo synapse. Metaphase, spindle fully forms. The tetrads are pushed to the middle, metaphase middle. So this isn't different, but the way that the chromosomes line up are different. And we're going to do a side-by-side comparison in a bit. Remember that in mitosis, that this one its other number pair would be here, and this one, the other number pair would be here. They would all, the sister chromatids would be lined up in a line here, not as a tetrad. So here you can see, this is the way the chromosomes line up in meiosis. This is the way they line up in mitosis. Anaphase. They unzip. So those proteins, those synapses proteins unzip. Half the tetrad goes one way, half goes the other way. A pair of sister chromatids goes one way, a pair of sister chromatids goes the other way. One pair of sister chromatids goes to one pole side of the cell. A pair of sister chromatids goes the other way. Remember, at this point in mitosis, what we were seeing was the individual sister chromatids were separating at their centromeres. One was going one way, one was going the other way. Here, what we have happening is a pair goes. So very different. Now, here's another weird thing that happens. A process called independent assortment, or what we mean shuffling. So that if we looked at one tetrad, let's say the first tetrad does this. They're just like, okay, we unzip each other, we've already exchanged our parts, and the top pair goes to the top, and the bottom pair goes to the bottom. Let's look at the next pair. The next pair could go like this. And the next pair could go like this, and then the next pair could go like this. And so they all don't just go, top pair goes this way, and bottom pair goes that way. They each do their own thing, where they either go like this, or they go like that. They independently separate which chromatid pair goes which way. They don't worry about what the first tetrad is doing or the second tetrad. They do their own thing. We could get tons of different combinations there. And again, why, if you have a sibling of the same biological sex as you, why you look different. Here's a bunch of different possibilities of what can happen. Anaphase 1, sister chromatids separate. You can see this pair, the purple went to the top, and in this pair, the purple went to the bottom. Telophase, kind of the same thing that happened in mitosis. But you've got sister chromatids together here on one side and sister chromatids here on the other side. At this part of telophase and mitosis, we had individual chromosomes on each side. So here's our process. At the end of this, we end up with two daughter cells. We were done at this point. When we looked here at mitosis, we were done. Mitosis did its job. It made two genetically identical daughter cells. But in meiosis, we are going to go do another phase, another round of phases. So these two daughter cells that are going to be made are going to go another round, and the sister chromatids now are going to separate in the next round of phases. What event listed below is one of the reasons for genetic variability seen in meiosis. The process of meiosis causes an increased mutation rate. Did we talk about that? I mean, it sounds interesting, but we didn't talk about that. Homologous chromosomes exchange DNA with one another. That sounds pretty good, right? Homologous chromosomes exchange RNA with one another. We did not talk about RNA. That's a distractor. Dividing cells exchange whole sister chromatids during meiosis 1. Did they exchange whole or parts? Okay, good. So B is the correct answer. Okay, meiosis 2. Meiosis 2 is the same thing as meiosis 2 is the same thing as mitosis, except we have two cells that are doing it. And those two cells have exchanged DNA. Okay, so prophase, again, same thing that happened here happens, but again, in two cells. And remember that our two cells did that exchanging of parts. So this stuff sticks. We don't go back to our original homologous chromosomes where they were. The changes, the exchanges, they stay. Just like when we exchange arms, we're going to keep them forever. We're not going to exchange our arms back again. We're going to keep our new arms. Metaphase, they line up at the middle. The spindle is fully formed. Anaphase, sister chromatids now separate. They do the same thing here as what happened in mitosis, is they go chop, chop, everybody on top goes one way, everybody on the bottom goes the other way. These do not do independent assortment. So chop, chop, top ones go to the top pole, bottom ones go to the bottom pole. No independent assortment happens in meiosis 2. Telophase, we start to get the pinching in. We start to get our nucleus reformed. Nucleolus comes back. Spindle's going away. Cells are pinching in and dividing. Cells are elongating. Okay, so if a diploid cell replicates its DNA so that it now contains an amount of DNA equal to 4n, 4 sets of chromosomes, how does a haploid gamete get a 1n number of chromosomes and a 1n amount of DNA? There are 2 meiotic divisions and 4 daughter cells produced in meiotic cell division. Did we get 4 cells, each with 1n amount? We did. There is only 1 meiotic division. That's not true. We know there's 2. There's 2 rounds. So, there's only 1. There are 2 meiotic divisions and 2 daughter cells produced. Are there 2 in the end, or are there 4? Okay, so A is correct. So here is our whole process of meiosis. We get that freaky synapse crossing over here. In prophase 1, we get independent assortment in anaphase 1. And our phases of meiosis 2 are similar to what happens in mitosis. Okay, so let's talk a little bit about the uniqueness of oogenesis in comparison to spermatogenesis and what we just described with meiosis. Spermatogenesis essentially does what I described as a generic meiosis. Oogenesis is going to do some other weird things, and we have a lot of reasons for that. The 2 O's sound like O-A, so you know for the future. Yes, we're going to talk about those. Okay, so regardless whether we're talking about male sex cell production, meiosis in the male, or female sex cell production, they are formed in the gonads. For males, that is the testes. For females, that is the ovaries. Okay, spermatogenesis, the making of the sperm. I put this first because it follows what I just described as the steps of meiosis. 2 phases. We end up with 4 equal-sized cells, each with an n amount of chromosomes, 1 set of chromosomes. So stereotypical meiosis occurs. Now, Okay, here's where you get the sexy sex ed lesson today. Sperm are tiny, teeny, teeny, weeny, microscopic, tiny. Female reproductive system. Okay, so there's the female reproductive system. Here's the vagina, the cervix, the bottom of the uterus, the fallopian tubes, ovarian tubes, oviduct, and the ovaries. The ovaries are where the oocytes or potential eggs are formed. Sperm. In comparison, the sperm are like smaller than that. I know a lot of you can't even see the little green dots that I made on the board, because that's even being generously large in comparison of the female reproductive system to the size of a sperm. So let's think about you. This summer, you're like, I'm going to get in shape and I am going to run the Chicago Marathon. And you, as you prepare all summer and the fall, would it be good in your training to take a backpack and fill it with every textbook you've had in the last 4 years and carry 2 gallons of water while you train and while you run? Is that a good idea? Simply, is it a good idea to carry all that stuff when you're going to run 20 miles, 26.2 miles? No. You're going to wear like shorts and a t-shirt, and maybe you'll have places that you map out. I'm going to get water here. I'm going to get water here. I'm going to get water here. You're not going to carry water, right? You want to be as light as possible. Sperm, they are made to be as light as possible. Sperm cells do not have much to them. Wait, I don't get that. Okay, so a sperm cell it has a tail. It essentially has 4 parts, and that's it, because it is stripped down everything, because coming from the vagina to get to the oocyte or the egg, it's going to run a marathon.
Do they know where to go, or do they just swim randomly?
That is a good question that I believe there's like detection of things that are released by the egg, but it doesn't always work with every sperm. So some of them go in here and die because they're like, I'm going to just go around, and they do a lot of like, and some of them end up over here. A lot of them end up over here. And only some of them actually get over here to the oocyte or the egg. So a lot of them die trying to find the oocyte. So because they don't really necessarily know where they're going, they go further than the marathon. It's like if somebody said, oh, you're going to start the marathon here and you're going to run for 26.2 miles and they don't give you a map, do you know when to stop? Not necessarily. So that's kind of like them. They don't have a map. They don't have it all mapped out for them. So a lot of them go even further. So they don't want all this stuff on them. So they are stripped down. They do not have all the organelles that are necessary to make a full cell. So guess who does? The oocyte has a full set of organelles. The sperm's job is to get here. It's got two jobs. To get here, one will dump off its DNA. Thousands will work together to release these enzymes in their heads as they all bump into the egg. They release these enzymes, weaken barriers. There are various barriers that allow, in which the oocyte allows only one sperm to get in. But there are thousands of them that need to get here to weaken the barriers so that one can actually wiggle through the layers. And then once the first one gets past layer 1 and layer 2 and into the oocyte's nuclear or the cytoplasm of the oocyte, then those barriers freeze and kill all the sperm that have wiggled their way into the outer layers. So there's some assurance that only one sperm gets through. So it's like crazy. Okay, so a little background on that. The production of the, I keep saying egg because we don't call it an egg until the oocyte is fertilized. Very few of our oocytes actually become eggs. So there's a huge strategy in oogenesis here because we get what's called the primary oocyte. And it goes through the first phase of meiosis. After the first phase of meiosis, what you see here is what's called a secondary oocyte and a polar body. What happens is because this, eventually this, is going to receive the DNA from the sperm, what it has to do is it's got to have the space to take the sperm's 1N amount of DNA and its own 1N of DNA, combine them together so it makes one cell. So it needs a lot of space. It needs a lot of energy to do this, and it also needs a lot of energy because fertilization occurs here in the fallopian tubes. Fertilization does not occur in the uterus. Fertilization occurs up here. It takes a week for a fertilized egg to make its way into the uterus and embed itself. It does not make a blood-to-blood connection with the mother for between 2 and 4 weeks after fertilization. So it's got to have enough energy here for almost a month to sustain itself and get energy from the endometrium before a blood connection is made with the biological mother. So it needs space and it needs nutrients. So what happens at the end of meiosis 1 is the DNA is halved. So here we get a 1N with sister chromatids. So it's 1N times 2, right? One chromosome with a sister chromatid. Instead of calling it 2N, we call it 1N because these are the same. 1N times 2, 1N times 2. Here's the thing about DNA. We know DNA is really, 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 really important, right? But if you have too much DNA, it's a bad thing. If you have too little DNA, obviously, It's a bad thing, right? So if you're missing a chromosome, if you are fully missing a chromosome, can your cell function? Depends on the chromosome, and most likely no, right? So too little DNA, clearly not a good thing. But if you have an extra chromosome or an extra piece of chromosome, what can happen is that your cell produces too much protein, lipid, whatever it's producing, that particular cell, and it can actually hurt you. So we want a precise 1N amount of DNA. That's why here we get this little thing called a polar body. And the polar body, it contains a set of DNA, takes that away, because we don't want extra DNA in our eventual egg. We want the right amount. A polar body is essentially just the chromosomes and a set of organelles. It doesn't have much space. Gives all of its cytoplasm over here to the secondary oocyte. In cytoplasm is a lot of energy. So this gets to be a bigger cell. This gets to be like just DNA and organelles. What our body does at that point, it absorbs that polar body. We don't need it. Just we use that energy back. Yes?
Would the Down syndrome be an example of the chromosome comparison?
Yes. Down syndrome, the person who has Down syndrome has three number 21 chromosomes. Having a complete extra 21 chromosome allows you to live, right? The people with Down syndrome live lives. They have heart issues. They have growth factor issues. They're often shorter. They have joint issues. They have mental retardation. They often have issues with immunity. There's a myriad of different issues that they have because too many things are produced that are good, but too much of a good thing when we're talking about what our DNA produces is not a good thing. So we want preciseness here. Then, this is controversial, not controversial, but this is argued upon that the polar body then goes on to divide, but most likely because there's not a lot of space, the first polar body is essentially the DNA and a set of organelles. Does it have the room to undergo a proper division again? Probably not. So some textbooks will show it like this. Probably just gets absorbed by the body. Meiosis 2 probably never happens. Secondary oocyte, here's a key thing. If this is fertilized, then the secondary oocyte undergoes meiosis 2. Then the secondary oocyte becomes an egg. But let's say that during ovulation in a biological female's cycle, the egg is ovulated, there is no sexual intercourse or no sperm that comes into the reproductive system. Within a few days, what happens is this starts to, when it gets to maybe about here, disintegrate. Because we don't need it. It didn't get fertilized. After a few days, the egg disintegrates, begins to target or to trigger, I should say, the cycle of menstruation about a week later. If this is fertilized, then, boop, it undergoes meiosis 2. It becomes technically an egg. This receives the DNA of the sperm. DNA comes together. Another polar body is formed. We only want the egg to have a 1N set of chromosomes or DNA. This takes half the DNA and gets absorbed by the body. So precisely clever. So perfectly orchestrated so that this can spend, a fertilized egg can spend the next week feeding itself off of all the cytoplasm that was received from this and that until it can get into the endometrium, and get more nutrients there, and then a few weeks later get a blood-to-blood connection with the biological mother so that it can be nourished directly from the mother's body. Amazing. Okay, so what causes genetic, so far of what we've talked about, what causes genetic variation meiosis? Does crossing over cause genetic variation meiosis? Yes. Should we stop there? No, not good test taking skills. Independent assortment? Wait a minute. Yeah, right? Does that one cause genetic variation too? Yeah. Okay, so we get two right answers. Just crossing over? No. Just independent assortment? No. Both? Crossing over and independent assortment. Okay. Okay, other variation that happens. Other genetic variation that happens. You've got a parent cell and a parent cell. And they bring their whole genome, their history of their parents and their parents' parents and the parents' 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 parents, right? And so they're bringing all of their DNA from their lineage, and you have another parent who's bringing all their DNA from their lineage. Those lineages can be very similar or they can be vastly different. Regardless, new allele combinations come from each side, and we get more genetic variation. When gametes fuse, there are 64 trillion different 2N combinations in a gamete. You are so unique. You are more unique than the ability to win the lottery. So if you think there's nothing special, I've had students, like, I do a discussion board in my online classes, and I say, tell me one thing that's unique about you. And some students say, there's nothing unique about me. I'm like, there's 64 trillion different possibilities of uniqueness in you. Random mutation that we've talked about, checkpoints, right? Each of those checkpoints, something could go wrong. Those mistakes that happen by chance, they cause new genetic variations. Here's the thing about mutations, is that mutations, we often think of like a mutant, right? If somebody called you a mutant, would you take that as a compliment, or would you be like, oh, now me, I would say thank you. That's right, I'm a mutant. Because mutations can often refer to good things, not just bad things. So mutations, whenever we have a trait like handedness, for example, that what we see over time is we probably started with one kind. Everybody was like, let's say, right-handed. And then eventually there was a mutation for a left-handed allele. Now we have two different possibilities over time. Eye color. We've got some basic eye color choices, but even that, I have brown eyes, and I know a lot of you I can see have brown eyes. Do we all have the exact same color brown? No. We all have different shades of brown, right? And each of those different shades is a new mutation. So we've got a lot of possibilities in terms of mutation. Mutations can be really, really bad, and which would activate our cells to say, kill it off. Apoptosis, not good. Our mutations could be no big deal. Is there, I'm trying to think of a trait, let's say hair color. Is there, like, just biologically, is there an advantage to having, like, in general, brown hair versus red hair?
Not today.
What'd you say?
Not today.
Not today, right? So maybe someday. But for now, it's not a big deal, right? You have red hair, you have brown hair, you have blonde hair, whatever color hair you have. No big deal, right? So there could be times of mutations. It's not bad, not good. It's just another option. Then there's some times where mutation can lead us to something better. So we have different shades of skin color, for example. I have very light skin, and having very light skin means that I don't have as much melanin, which means that I don't have as much natural UV ray protection in my skin, which means that if I go out in the sun for more than 20 minutes, I start to get burnt. That's bad, right? Some of you have way more melanin. You have much more natural sun protection. So maybe you could be out in the sun for 2, 3, 4 hours before you start to get burnt. Is that an advantage? Yeah. So sometimes mutations can be bad. Sometimes, just another option, or sometimes they can be good. Yes.
Aren't traits like the hair and eyes, eye color, not necessarily genetics, because they take more than 2 years to an exact phenotype of trait?
Yeah, there's some genes are one trait or 2 genes for one trait. Most of our traits in our body are like 30 genes to contribute to that one trait. So there's a lot of different possibilities, hence why eye color, that we could all get all of us brown-eyed people together and find nobody in the room has the same shade, because there's like 30 genes that code for just the color of your eyes. That there's a lot of variation. Genetics, we will talk about very simple means of genetics to give you a basis. But if you go on to take a genetics class, which I hope you all do because it's so cool, that you will find genetics and the way we express our traits is so complicated and so cool. And just, it'll be mind-blowingly amazing. Okay, so here's just like a lineup of, these are really good things to like go over by yourself, to sit down and say, let's look at a feature of mitosis and meiosis, a general feature, and let's see what the result of it is in mitosis versus meiosis. Those will help you get these ideas straight in your head. So star this to go over and make sure you understand all of these. Same thing here. The phases of mitosis and meiosis, make sure you understand that the first phases of mitosis are wildly different, but very similar to mitosis. And the second stages of meiosis are very similar to mitosis, except for in meiosis, we have 2 daughter cells undergoing the similarities there. Okay, so let's talk, lastly, just a few terms here and there to discuss stem cells versus differentiated cells. Okay, so as we get here, this first cell, when you have a fertilized cell, what happens to this cell, the zygote, is that cell splits in 2, and it splits in 4, and then 8, and then 16. And it undergoes what we call cleavage, and it keeps cleaving, and you get a bigger and bigger, you get a ball of cells that stays about the same size for 2 weeks, but the cells begin to get more numerous and smaller in size. The first 2 weeks, when we're talking about stem cells, the first 2 weeks, we call them embryonic stem cells. They are the most potent. Pluripotent means that they can become, in the first 2 weeks, any kind of cell in your body. I'll get to your question in a second. These cells are not programmed to become anything specific yet. So potency is a big deal in embryonic stem cells. There's a lot of controversy about the potency of stem cells in this first 2 weeks, because I think we can all agree on this. If we could take a ball of cells, and we could take that ball of cells and we could program the millions of cells by the end of the second week to become cures for millions of people, would you all be cool with that? Would you be like, yeah, that'd be great, right? Can we just agree that that would be great to cure people of bad things? Okay. But here's where the controversy comes in. That ball of cells, even though it has the potential to cure other people, you take away the destiny of that ball of cells to become a person. And that's where people say, no, do not touch the embryo. That's why we have all this controversy about abortions. I'm not saying one side or the other. I'm just laying out the facts here. That ball of cells could potentially be someone who cures climate change, solves it, who becomes the best world leader on the planet, brings everyone together, stops war, stops famine, or they could be a total dirtbag, right? They could be a person who goes to a mall and shoots 1,000 people. The destiny, we don't know. But I'm just laying out for you that you've got these two possibilities in that first 2 weeks. You've got a lot of potency of those stem cells to become whatever somebody wants to get in there and program them to be, or you take away the destiny of that cell. After 2 weeks, that ball of cells, those cells start to commit to being this, or this within a body. So they lose potency. When you get to be an adult, we have very few stem cells left in our body, true stem cells. We have some in our brain. We have some in our circulatory system. So we have less potency. So our potency, 2 weeks, really, like sky's the limit. After that, it goes down into 3 categories. And a couple weeks after that, then cells start to commit to being very, very specific things. And adult stem cells, we have very few that can do anything like, but they do have to stay in a category. Now, somebody, or I would say a team of people, figured out that some adult stem cells can be programmed to act like embryonic stem cells. So somebody figured that out. So then that negated the need to work with embryos. So it takes away that, controversy there. Differentiated cells are when a cell commits. It's like, I'm going to be a heart cell, or I'm going to be a pancreas cell, and I'm specifically going to be an islet cell that makes insulin. So your commitment or your differentiation continues. I'm sorry, you got to speak up. No, just this, yeah. Okay, so embryonic stem cells, as I just mentioned, highest potency. Adult stem cells, the potency, it goes down to being like circulatory system related or brain related. But we can manipulate some of those to have higher potency. Germ cell produces gametes. Gonad are the organs that produce a gamete. So just the difference in terms. Okay, a chimpanzee has 48 chromosomes in its somatic cells. How many of these chromosomes are autosomes? Okay, shout it out.
46.
Good, very good. 46 autosomes and 2 sex chromosomes. For any eukaryotic cell, there are 2 sex chromosomes and the rest are autosomes. Okay, 2 sex chromosomes, rest are autosomes. Make a note, you do not have to do objective 6 and chapter 11. It says briefly compare the